So I think we'll just go ahead and get started. Today we have Adam Sigler, MSU Water Quality Associate Specialist, and Hannah Riedel?
Close.
Riedel?
Rhymes with beetle.
Okay, Riedel. Okay. From the State HAB Program Coordinator from DEQ. And they're going to talk to us about some harmful algal blooms and some best practices, risks, and resources. And so I'll turn it over to Hannah and Adam, and then at the end we'll have some questions.
Thank you, Megan. Maybe I'll just start off for a second. Hannah's going to do a lot of most all of the talking today. We picked this topic because we've been over the years. I've got a handful of questions each summer about various algae topics in stock ponds or in streams or other water sources, and was just talking with Hannah about the fact that she's been working on the HABs project for 3 years now. So I've been working with her over that time, and she spends a lot more time dealing with algal blooms than I do. So I was happy to hear that she was willing to come on and join us for the conversation today. And so I guess I'm glad that Megan did the introduction so that she could mispronounce Hannah's name instead of me, because I would have pronounced it wrong, which I'm embarrassed about because I've known Hannah for a long time. So it rhymes with beetle. That's helpful. Riedel. So I'll just tell people where we're going, and then I'll let you do maybe more of an introduction and roll through. And are people seeing? I'll assume that folks are seeing Hannah's slides okay. I'm seeing them okay. And I'm going to try and monitor the chat while Hannah's talking. So if people have questions and you want to type in there, that's great. And we definitely want to open it up for questions and discussion at the end. We have a couple of topics that we've lined up in advance to talk about anything that y'all want to talk about related to harmful algal blooms, which are a specific type of algal bloom that Hannah will talk through. And then she's going to go through the existing, so some basics about harmful algal blooms, what they are and what some of the concerns are with those, and then existing resources, largely on the website that Hannah helps to maintain as part of the state HABs team. And then she'll walk through the website for reporting of potential HABs, which is a really great resource that we have, that Hannah is a primary manager of here in the state. And then some basics on testing, which has been important for people over the last couple of years, and opportunities for extension to serve as locations for test strips, potentially for toxins associated with HABs. So we've got an opportunity there we can talk about. And then discussion. We've got a handful of questions, so be thinking about your questions, and if you want to type them in during the chat, we can do that. Or I think we can probably also keep it unstructured, and if you've got a pressing question, then you can jump in at any time, I think. So, and just unmute and do that. So with that, Hannah, I'll turn it over to you and let you walk us through these points.
All right, sounds good. Thanks, Adam. Yeah, thank you for the introduction. I work with the Department of Environmental Quality, and I'm here today representing the state harmful algae bloom program, but I also work in our non-point source program at DEQ as well. If you have questions about that, I'm also a good resource for that program. But like Adam laid out, I guess the main takeaways I'd like you to get from this presentation are understanding what harmful algal blooms are. If you get questions from landowners, for example, concerns about HABs, what resources are available to you to help answer those questions. And then just kind of seeing how you guys feel about the potential you can serve as a really important liaison between landowners and DEQ. As you all know, we have a large state, and it's really kind of just me responding to reports around the state. So the more help on the ground locally I have, the better, more effectively we can respond to these reports. So to get right back to the basics, I'm sure you all have a pretty good understanding of algae, but nutrients are a really essential component of life and freshwater ecosystems, especially. They grow algae, which feeds fish and animals. But when you have too many nutrients or too warm of water, it can lead to a proliferation of algae growth. These images I have on the screen, these are photos of green algae. So there's many different types of algae. For the purposes of this talk, we can think about green algae, which is your quintessential like filamentous algae. This photo in the lower left is from the Smith River. It's been seeing a lot of green algae blooms. Same with the photo in the center. And also the photo on the lower right, that like clumpy green stuff that's generally growing up from the bottom of that pond. Green algae does not pose any potential health risks, but it can definitely be a nuisance for recreating. It can stink, and you'll often see fish kills associated with any type of algal bloom because the decomposition of those cells depletes oxygen in the water. So often when you see fish kills in algae, it's because of oxygen depletion, not necessarily toxins. Another type of algae, also known as cyanobacteria, actually are pretty visual of cyanobacteria. The green, sorry?
You're breaking up pretty bad. Like not your voice, like you're cutting in and out really bad. Maybe turn off your video and see if that, yeah, the bandwidth is, I don't know. There's been a lot of issues lately, I think, with that.
Do you think I should back up a little bit?
I'll try. Yeah, maybe when you first started the Habs 101 slide.
Yeah, just this slide it started to break up on, I think. Maybe okay. Matt, you kill your video too?
Thanks, guys.
Thank
you. Okay. So I was describing green algae. It commonly grows from the bottom or the substrate of a stream or lake. Like this photo on the lower left is from the Smith River. Same with the one in the middle. And then the one on the lower right is green algae growing in a pond. And yeah, generally this algae will be somewhat attached to the subsurface or the bottom of the water body, or it will grow really long and stringy. Green algae is not going to be a health concern, but it might be a nuisance. Blue-green algae, another word for harmful algae blooms. The photos I have on the screen now are really common characteristics of blue-green algae. On the lower left, you can see it's kind of like pea soup. There's some blue streaks and gold. Essentially, like really bright colors are an easy characteristic to identify blue-green algae. Photo in the center, the blue-green algae looks like kind of like grass clippings. And the difference between this and the filamentous green algae, it won't be growing attached to the bottom of the water body. And then this image on the right, that's another characteristic where it kind of just looks like someone's dumped a bucket of paint in the water. I'll probably throughout the talk refer to HABs as cyanobacteria. And also known as harmful algae because the cyanobacteria can produce cyanotoxins. And the toxins are what pose the health risk. So I'll repeat this a lot throughout the talk, but you want to avoid coming into contact with a harmful algal bloom. If you're ever uncertain, the best practice is just to avoid it. Don't touch it, don't drink it, consume it. Keep your pets and children away from it because they're especially likely to just blindly run and jump into a water body. And so if you think you or some animal or child you're concerned about has come into contact, just shower immediately. If you start exhibiting symptoms, call your doctor immediately. This shot up just to give a little historical context. This is a newspaper article from Canyon Ferry in 1904, and it's talking about poisonous algae on Canyon Ferry that killed cattle. So this is definitely not a new issue. These cyanobacteria are native organisms. It's just that our understanding of the implications of cyanobacteria blooms are a better understanding. And us humans have also created environments that are more conducive to growing cyanobacteria. So that has to do with things like increasing water and air temperatures, increasing nutrient pollution, and creating more stagnant water on reservoirs. So in Montana, we typically see these common types of cyanobacteria: Anabaena, Microcystis, Aphanizomenon. And commonly, these cyanobacteria produce a certain type of cyanotoxin. So commonly the toxins we test for in Montana are microcystin and anatoxin. I am not I work for DEQ, so I'm not an expert on human health issues, but some common symptoms related to exposure to these toxins are things like ear, eyes, nose, skin irritation, abdominal pain and diarrhea are also some common symptoms. Especially in dogs, you might see seizures or extreme lethargy. And I just want to point out too that a lot of these symptoms, like eye, nose, and skin irritation, are commonly co-occurring with things like allergies, or maybe your dog got into a lot of trash. So that's why one of the main goals of the state harmful algae bloom program is just making sure people have a resource to educate themselves on what to look for, because if you can identify potential cyanobacteria or know enough to be pretty confident it's not there, then that can help you narrow down different causes of animal or human illness. So monitoring cyanotoxins is really complicated and difficult. This map of the United States I have on the screen is showing locations where the different cyanobacteria blooms occurred, and the different colors just correspond to increasing severity. So obviously blooms are really common, and this is definitely not an exhaustive map of all blooms. But when water quality samples, well, let me back up. These points are showing cyanobacteria cell density. So it's not measuring the level of toxin, just how much of the organism is present. And when water quality samples were taken to assess how much toxins were present, that's what this map is showing now. There were a lot less detections of toxins. So that's one point I want to make is just because a bloom is present doesn't mean the toxins are as well. Just means that there's that potential for it to occur. And likewise, just because a bloom has dissipated doesn't mean the toxins have dissipated. So toxins can remain in the water for about a month longer than after the bloom has dissipated. So it's really important that you have a water quality sample if you want to confidently say if toxins are present. I always show these two photos when I talk about this program. The image on the left, there's an accumulation of that grass clipping like algae just right along the shoreline, but it looks like you could take a running jump and get into some nice, cool, clear water. Whereas the image on the right looks really nasty. There's that bright blue characteristic of cyanobacteria. But when water quality samples were taken from these water bodies, the microcystin toxin level was really high in that water, that 82 micrograms per liter is about 10 times higher than the recreational guidelines to avoid that water. Whereas the really nasty water, the microcystin levels were an order of magnitude low, the point at which we would start to be concerned. To further complicate monitoring for toxins, not only can the predictable when it produces toxins, but natural factors like wind can really change the distribution. So these two time-lapse photos were taken only 15 minutes apart. So this a common motto of the program is, you know, when in doubt, just stay out. I guess I'll pause.
I haven't seen any questions in the chat yet.
Does anybody have any questions for Hannah before we move on?
Okay.
Curious how many people, if you don't mind, either raising your hand. I've never actually really used that feature in here, but or typing in the chat if you've had a producer or somebody come in and report an algal bloom issue in your county. I'd love to get a feel for that. I've interacted with some of you on it, but I'd love to get a better feel for how many people have been dealing with these issues, maybe just in the chat.
Looks like a lot of people are raising their hand.
There is a question from Jane. Does toxicity vary based on animal? She's thinking about livestock producers.
That's a good question, and I'm guessing it probably would. EPA has set guidelines for most cyanotoxins based on recreational advisory levels and drinking water levels for adults and children, but they haven't set guideline levels for different types of animals. little bit later that those recreational guidelines are a higher concentration than the drinking water guidelines, and that's in part because we're assuming that your long-term effect if you're drinking water. So I think the guidelines that they've set based on recreation and drinking water would sufficiently inform your livestock. Helps answer your question. Curious for the issue that was pointed out in Blaine County, was that last summer on Cow Creek?
Oh, there's comments going just to Hannah, so I can't monitor all of them. I think. Seeing that comment. Oh, wait, we've had issues in. Okay, I'm sorry.
Julie, any specifics on the Blaine County there?
I just missed that one, Blaine County.
Julie's on, but
That's okay.
she's on mute or typing, so.
No worries. Yeah, last summer we had quite the issue on Cow Creek in Blaine County. So, it's amazing to see how many of you are familiar with this issue. So, I guess the next topic was going to cover what resources are available. I guess before I start diving down my slides, I encourage you to visit the have.mp.gov webpage. I'm going to show you that webpage in a minute here, but you can submit reports of suspected or confirmed harmful algae blooms there. And that's really beneficial because it helps keep members of the public aware of what's going on. And just as the state's environmental quality agency, we're tracking where those reports come in. And so that's useful for informing our assessments. And then I'll cover here in a minute too, I might be able to help getting resources to those livestock owners. I saw a couple of comments where people mentioned having cyanobacteria in stockwater ponds. So we want to help make sure, there's a resource for those concerns. All right, so the state HAB program, it's actually a collaboration between DEQ, Department of Health and Human Services. They're the ones who are best at answering questions about the human health impacts. And we also collaborate with Montana Fish, Wildlife, and Parks. And our main function, like I've said, is communication and education with the public. So I'm going to switch screens. Can you all see? Maybe a raise of hands if you guys are seeing the web page pop up.
It's loading, I can see it.
So I just typed hab.mt the search bar. The web page changes, but that's just a really easy one to remember. And the hope is the web page is a really useful resource. So the first thing I want to point you to is this link here to report a possible harmful algal bloom. It's really easy. The only information I think it requires that you put in terms of location is what county you're in. So if owner is sensitive about the location of their property, that's fine. We can just here. A really helpful pieces of information are contact information, so I can follow up, especially if there is a question or concern associated with the report. So you can briefly describe the bloom. We have questions like if it's been seen on this lake before, if it's publicly accessible, that's important because, again, we want to make sure other members of the public are aware. The name of the water body is required. If you don't know, you can just type some sort of descriptor in there. If there's an illness, that helps us kind of prioritize our response. I can get really busy in the summer sifting through these reports, but I still encourage you to contact a veterinarian or a doctor if there are serious symptoms going on, or even minor. Just once symptoms start occurring, reach out to a professional who can help you there. There is this map, so you can geolocate the bloom. This is really helpful too, especially if you don't know the name of the water body. It helps me identify like what landowners there are, if it's a public landowner. And then also, most importantly, is this function to upload an image. Visual assessment is really our first tier of monitoring. We can tell a lot by looking at a photo. If it's green algae, if it's obviously blue-green algae, if it's not so obvious, then we can follow next steps. So it's a really reporting platform. You don't need to download an app to access it. You just go to that hab.mt.gov webpage. We also have a slideshow of photos to help teach people what to look for. There's tons of information, like quick videos, from both DEQ, EPA, CDC, some questions, ask questions directly on the webpage. I want to quickly walk you through this guidance document. That's just down here on the left-hand panel. There's a thumbnail of the title page. But this document is, it's intended for people who have jurisdiction over water bodies, so maybe like a state park manager or even a private landowner who has land around a water body. There's just some background and pictures to look for. Green algae photos across the top, blue-green algae photos across the bottom. About exposure pathways and some human health impacts. Here are EPA's health advisory levels for drinking water, and then we also have advisory levels for recreational water. So I'll point out here, there's this cyanobacteria cell density, so just a measure of how much of the organism is present. I said before that toxins are not always associated with the organism itself, but it can be a good predictor. So a really high density might advise closing that water body or a portion of the water body. And then we kind of have these two cautionary tiers. We used to have this middle tier be a warning, but we felt like it just required a level of monitoring that was not feasible to distinguish between those two categories. But so these toxin levels can be a good reference. And then we have recommended actions for the different measures. So commonly we're posting signs helping make people aware of the situation. In a worst case scenario, we're closing those water bodies. Once you get to this middle tier where like microcystin is above 8 micrograms per liter, that's really where keep livestock away, for example. These levels are less than 8, so that's just basically a presence of cyanobacteria. So like I was saying, conditions can change really rapidly, so you might want to be careful, but based on existing information, there's no reason to be concerned based on these numbers. And we also have this flow chart, which helps walk people through kind of different steps you would take for monitoring a harmful algal bloom. So we start with a visual assessment, just looking at photos, basically. The next step is test strips, which I'll talk a little bit more about later. These are quick, relatively quick. You can do it in the field. They're pretty easy to follow. So you can get a good amount of information pretty quickly using these test strips. If there's animal or human illness that's confirmed to be associated with a cyanobacteria bloom, then we generally would kick this to a more extreme level of caution. I will say that closure is something that like a county health department needs to administer. DEQ doesn't have that authority, but sick illness or animal death would definitely kick us more to this lab testing. So the test strips are semi-quantitative, whereas lab testing is fully quantitative. You're going to get right at that level that would inform you whether you need to close the water body, keep an eye on it, or definitely keep livestock and people away. And then there's just a little bit about the visual assessment, field tests, laboratory analysis. So I'll talk more about monitoring methods in a second. The last thing I wanted to point out about the webpage was access lab reports. I'm not going to take you there right now because to my knowledge, HAB season has not fully gotten underway, so there's not a lot of reports to see on that map at the moment. But I'll show you kind of what this is a screenshot of the map taken towards the end of last summer. So the red dots show reports that were more recent, and then the green to purple dots show kind of all reports in our database. So keep in mind, these are just citizen reports from folks like yourselves or recreators. So it's definitely not an exhaustive map, but it can be informative for other people in the state. Adam and I and others at MSU Extension have been working articles on this topic in the Lives in Landscape magazine. So this article was published in your spring edition, and then we're working on another one for the summer edition. So that's a resource there. Those will be linked on the hab.mt.gov webpage here soon, I hope. Okay, so diving into those different monitoring resources. So if you submit a photo to me and I say, gosh, that looks a lot like cyanobacteria, there are a couple options that landowner could take. They could decide they just want to use utmost caution and avoid the water until that bloom's dissipated for a good amount of time. If they need more information, our program purchases a batch of these Abraxas test strips the beginning of around this time every year. So this is a picture of kind of what it looks like. It's literally just a test strip. You grab a water sample in any kind of bottle, really, and you add some reagents that come with the strips. Do some shaking, do some weighting, some more shaking, and about 45 minutes later, you'll have a presence or absence result. So the photo on the screen, this is a microcystin test strip. We also have test strips for anatoxins, but you can see there's a control line and a test line visible. So it tells you there's no to very levels of microcystin present. So this 10 parts per billion microcystin, that's around that recreational guideline when you really want to start advising people and animals stay away. So these are nice because they can get you good information relatively quickly. If you didn't have a test line there, you would know there's cause for concern and increased caution. But these are limited because you can't get a true measure of concentration. I'll just point out that the state HAB program purchased these and distributes them around the state. In the past, we've distributed them to FWP's regional offices, but they're also available to the public. It's about $30 per test, and you get a pack of 5, so it's like $100 to $150 for a box of, say, microcystin. So somewhat pricey, but it's available. Got a positive detection, and I would say especially if the water body is used for recreation, then that county sanitarian, for example, is probably going to want to know a good idea of what the concentrations are so they can determine whether or not they need to close that water body. So our program works with the EPA Region 8 lab. It takes about 1 day for them to overnight a sample kit. Has everything you need to collect the sample, package it back up, and ship it back overnight, back to the lab. And then it takes 1 to 5 days to get your results back. So this option does not give you speedy results, but it gives you accurate results, at least for the time at which that water sample was taken. And there are private labs in that guidance document I mentioned. Provides reference to some of those private labs, but our program works with Region 8 labs. I saw some questions pop up, so maybe I'll pause and get to those.
I can read out a question to you from Shelley Mills. Is there a use by date on the test strips? She's purchased a box for their office.
Yeah, so the test strips expire pretty quickly. I think they're only viable for 6 months to a year, so that's why we always purchase them around this time of the year.
It's a bummer that they're so short. Otherwise, it would be nice to be able to get them into more offices, but
I know.
short expiration periods, it's hard to
I
justify.
guess the nice thing about expired kits is they can be a nice like educational demonstration if you want to take kids out and use them there.
That's interesting. I wonder if it's I wonder about their efficacy. Like I have some bacteria enumeration media in my lab that and some of it's like 10 years expired. And last year I did an experiment to see whether the age of the material was correlated to how reliable it was. And it was. The old stuff was the least reliable. So yeah, it's interesting to think about whether you could use them for education and you might still get a positive even on the expired ones. Obviously, you wouldn't want to rely on them though.
Okay, so I'm almost through my material, and then I think we mostly just wanted to hear from you guys. I was going to point out this kind of other tier of monitoring is using satellite imagery. This is really neat because you can monitor water bodies remotely. This is a software that EPA, USGS, and NOAA created where every day they're uploading satellite images, and they basically take the spectral signature from a water body and convert that to a density of cyanobacteria cells. So I just changed the screenshot to show Canyon Ferry at the end of 2018. So the red colors show high density. So this is neat for monitoring remotely. If water body we can't easily get to, we can kind of check and see what's going on here and determine how high of a priority it might be to do some on the ground monitoring. But this is limited because it has a really high error rate. Things like clouds and potentially, apparently, even like glacial runoff can really, it kind of triggers a signature of cyanobacteria. And this software is only applicable for large lakes because each of these little pixels is like a 30 by 30 meter cell. So not extremely resolute, but it is a resource I just wanted to put out there if people have more interest in it. So yeah, I think here we were interested in hearing from the different extension offices. I mentioned our program distributes those test strips to the different FWP regions, and it looks like Adam has kind of pinned the names of the cities where those test strips go onto the map of your extension offices. But I would be interested to hear from each of you. Have you dealt with this issue before? Are you interested in getting your own batch of test strips, and are you comfortable using those to respond to a report?
It's okay. It sounds like an interesting opportunity we have here where Hannah has said that they've distributed test kits to all of these yellow labeled towns, but and so are all those at the Fish, Wildlife, and Parks offices, Hannah, or some of them other locations?
I think the only one that's not is Whitefish. They have a monitoring program that covers like 50 different lakes. So that
Oh, right.
that Whitefish is actually a nonprofit's location.
Gotcha. So it sounds like there's an opportunity to try and, I know we corresponded with Rose and Cascade last year. Gosh, was that just last year? It seems like an eternity ago. Um, and there was a cattle mortality, and they wanted to confirm, and there was some struggles with getting test strips in a timely fashion. And so in Great Falls, that there's typically the Fish, Wildlife, and Parks office there, but in other locations, there's some gaps in the state, and maybe county extension offices could fill those gaps. If folks are interested, we'd love to hear about that.
Julianne, where are you?
I'm in Blaine County. I'm in Chinook. I'm the Cow Creek Reservoir last year.
Cool. Okay, great. I'm going to make note of that.
Haver.
Shelley asked a question, and I'd like to know too, how long are the strips viable for? And if we did want to participate, would we be purchasing those ourselves, or will you provide them to us? Or
So those test strips are viable for, I think like 6 months to a year. So we typically make our order in the spring so we can cover the entire growing season. The question we're asking you now is if you would like DEQ to provide them to you. So we have resources allocated to distribute these around the state. So it would be at no cost to you. I guess I brought up that they were publicly available because I know some landowners say they have property right along the shore of a lake and their grandkids come to visit. They want to test really frequently, and that frequency doesn't really work with how we've budgeted for these resources.
So it seems like, I mean, we could, Hannah, correct me if I'm wrong, but could identify some counties that are interested in getting them and could send them out. And then if those get used, then they could be replaced within the year. And it might be a thing where, I mean, I was kind of thinking about, are there counties where we have people going back and forth regularly where we could move test strips around when it comes? Because what did you say, Hannah? It's 3 tests that would be in each county.
Yeah, so you order them from the company and they come in kits of 5. Last year we split them up into batches of 3. So I think that was a question someone had if we could just split up the box. We can definitely do that, and that's actually what we've done in the past. I think we at DEQ would just want to do that because we want there are a number of different this is not the right word, syringe, something like a syringe, different vials, syringes, and we just want to make sure all the parts get distributed appropriately. But we can think about splitting them to different counties, yeah.
Shelley Mills has a question about whether we're suggesting a routine monitoring program or just in response to requests.
Yeah, this would be just response-based monitoring. I think if your county and other partners in your county have interest in routine monitoring, I know in Beaver Creek outside of Havre, I think that's MSU Northern there, right? One of the professors there has a research program going on, so I'm happy to help think about routine monitoring, but for the purpose of distributing these Abraxas test strips, now we're just thinking about having resources to respond.
Yeah, that's how I've been thinking about it as well, for sure, Shelley. I think that they're so expensive that thinking about routine monitoring seems, unless there's an issue or a water body. I had a sounds like I asked the question in the chat whether producers who have reported algal blooms had a place to move the cattle off. Because so you see a bloom in a stock pond, I mean, the first step would be to submit a photo to see whether or not it looks visually like it's cyanobacteria versus green algae. And then if it looks like it is cyanobacteria, then you could do a toxicity test. But the problem is that you can't predict when they're going to release toxins. And so if there's cyanobacteria there could be toxins not here today, and it could be here tomorrow. So there's risk. And as soon as a cyanotoxin bloom is found, the producer wants to pay attention to that, because you can lose animals in a hurry if the bacteria start to release the toxins. And so, I mean, ideally, if people can think about what their backup plan is, if a stock pond experiences a bloom, then they can have that plan in place. And it sounds like there was one instance in Yellowstone County where a producer did fortunately have a place to move their animals to. But that's I mean, it's not always going to be the case. And so that's why we worked with Tommy on writing that. I worked with Tommy Bass on writing that article about stock ponds specifically, to reduce water quality issues in those stock ponds, specifically to reduce phosphorus runoff. because cyanobacteria are nitrogen fixers, and so they can fix their own nitrogen out of the atmosphere. And so I think that there's still going to be some nutrient limitation likely, but just conceptually, it makes sense to really focus on trying to keep phosphorus out of these water sources. Definitely phosphorus and nitrogen, but yeah, so I'm really just trying to think about how to help producers be proactive about addressing these issues and then having the resources in place to do this test strip testing when it's necessary, or when a bloom does happen, if they can't move the animals someplace else.
So Tim Fine kind of had a question, I guess. He said he'd be interested to participate, but the concerns that come into your office vary so much year to year. You'd be concerned about wasting the test strips. I would say don't worry about that. We're not sending dozens and dozens of these to individual offices. It's 3 to 5 tests. So I think just like people have been saying about coronavirus, it's better to be more prepared than less prepared. So I think having these distributed, especially to places where you've gotten questions on this topic before, is worthwhile.
And Hannah, correct me if I'm wrong, but y'all at DEQ are buying a set number of these, and so you've got them, and they're already purchased and they're already going to expire. And so the thing here is that if we can get them spread around the state a little bit more so that they're more readily delivered when the issues come up, then they can get used quicker and more timely. The DEQ's already made plans to buy the number that they're going to buy, so that they're going to expire wherever they're sitting if they don't get used. And if we're able to just have them better distributed, we can be more timely. So Shelley has said that she could do that. Valley County. And
Tim, where are you located?
Sydney.
Sydney, okay. Yeah, and I think I saw a comment was that from Rose, that she had gone to the Great Falls Rose said she went to Great Falls and FWP, didn't know what she was talking about. Again, another example why it's nice to have these distributed, and I'm really glad to be here talking to the extension agent now. So we work with the fisheries biologists at FWP, so there's a chance, I guess, maybe you didn't talk to that FWP representative. But also, especially this year, I think FWP is going to be trying to step back a little bit and really only respond to reports on their fishing access sites or state parks. So that's kind of another factor going into this discussion. Colleen, I see your comment too, so I'll make a note of that.
Ben's got a question here about research showing the effectiveness of barley straw on reducing algae blooms, and I haven't been on that in the last couple of years, but I worked on an article on that a couple years back, and there was the research that I found at that time was anecdotal. It was people had done, they hadn't done really controlled science on it the last I saw. But the anecdotal work suggested that it could suppress growth.
Yeah, I think there's more research coming out on that approach and other mitigation measures now. Barley straw could be effective, especially if you're thinking about a really small livestock tank. I think when you start thinking about reservoirs that are like 30, 50, 60 acres large, you're thinking about applying a lot of barley straw. And it's my understanding that the timing of adding that to the system needs to be kind of just right. You have to do it well before the algae bloom starts appearing. And typically you have to reinstall those barley bales multiple times in the summer. So it can just become a lot of labor. And then I get a lot of questions about all these different mitigation measures that are coming about. And the general principle I try and remind people about is that all of those approaches aren't really solving the problem. The problem is often like nutrients are getting into that waterway. So unless you cut off that nutrient supply, then you're going to need to continue with that labor intensive strategy. Another problem with barley straw and other mitigation measures is they're just adding more nutrients into the system as that barley straw begins to decompose. So it can be a solution, but it can also not be. I always advocate to try and focus more on making sure you've done everything to eliminate sources of pollution before you start investing resources in measures like that.
Tyler Lane asks about whether the blooms are more prominent during drought. And I'll take a stab, and then they will respond to temperature. And I think temperature can sometimes be associated with drought, I guess. So I don't know if there's a direct correlation, and it's probably going to vary on the water body whether during drought you have worse water quality and warmer temperature or not. But I'd say that it's very possible that they're worse during drought because the water gets warmer and the algae are happier. Does that make sense to you, Hannah?
Yeah, I would agree with that. Just more anecdotally, we've seemed to receive a lot more reports, or at least locally, the like Canyon Ferry and Hauser Lake chain of lakes that I live near. We see a lot more persistent blooms when there's not bad wildfire smoke. So having those like nice, long, clear, sunny days can really promote growth of blooms too. So there's just a lot of factors that go into it. If it's not windy for a long period of time, you'll often see a lot of blooms then too.
I wanted to ask people if they see obstacles to folks, producers maybe specifically submitting reports to the HABs page. I feel like it's such a good resource to have folks like Hannah looking at pictures to be able to confirm whether or not it could be cyanobacteria or if it's green algae. And I think it'd be great to get more reports in there, both so that people are informed about it, and also because if we get more data about more reports around the state, it's going to help us to get more resources to address these issues. But I can appreciate that there's reasons why producers aren't going to want to put a report in. But so if all the information you have to put in is what county you're in, I feel like that could remove some of those obstacles. And I'm curious whether any of the agents have ideas about that, whether they see obstacles to people submitting reports with photos and how we might work around those issues. We're right at 3 o'clock, Megan. I don't know if anybody that's interested can hang out for a minute or we follow up on email, or I don't know how you manage that 3 o'clock hard stop. I definitely would be interested in anybody's thoughts on obstacles to reports. Another question that when we can wrap it up here, maybe if people don't have more questions, is the livestock indemnity program. I'm curious whether folks have worked with producers on the livestock indemnity program. One of the reasons for getting a confirmed test, we were working last year in Cascade County to try and get a sample sent in, and to confirm that there were toxins present after some cattle died. And it was largely for the, I think that the producer was thinking about the livestock indemnity program and getting compensated, but it seems like there's a lot of hurdles there. And we were just curious if people have successfully worked with the producer to move through that program.
So Adam, I know the LIP program for sheep hasn't been very successful. You'd have to have a horrendously high die-off. I think they recently changed the program. It used to be based off a national average. Montana has such a high percentage lamb crop that basically the national average, they use the national average lamb crop as the threshold. And since Texas has like a 75% lamb crop and Montana has a 125% lamb crop, we needed to lose like half of our lamb crop in an event before we had qualified for LIP. So most sheep people have kind of given up on LIP anyway. I think cows, it would be a little easier because the national average calf crop is pretty similar across the board. Although I did hear they just changed it to a state average. So.
Oh, interesting. Interesting. That was Brent, right? Thank you, Brent. Well, I'm available, always available if folks have questions. Please send them my way, an email or give a shout. And I know Hannah's got her contact information up there and is receptive. We'll see how things go this year, but I think that the trends in a lot of places show that the algae blooms are increasing over time. And places where it's being studied more effectively, they're really finding those trends. So, and Dave Brink turned me on to an issue related to this that I hadn't even thought about yesterday, and how if you're irrigating with water that's got cyanobacteria in it, you can have soil toxicity issues, and so there's some other things that I don't think we need to worry about yet, but that might be coming down the pipe at some point. So, thank you all for your attention, and I'll hang out for a minute if anybody has questions, but otherwise I think we can call it there.
Yeah, thank you, everybody. I just noted down Julianne in Chinook, Shelley in Valley County, Tim in Sydney, Colleen in Plentywood, and Molly in Hardin. So I'll follow up with you guys, getting you some test strips here soon.
Great. Thank you, Hannah, for talking today.
Oh, and Rose, gotcha, in Great Falls.
Thank you, Megan, for hosting this and pulling it together. Sorry that we weren't able to see all at Agent Update, but it's great to have this opportunity to share this info.
Thank you, Adam and Hannah, for providing this for us today. I think this was great information. That's basically all we have for this week. Thanks, everybody, for the great discussion. I know we don't have anything planned for the next couple of weeks on our Telework Tuesdays, and I know everybody's kind of slowly getting back to work, but I think if anybody has any specific topics they would like to really hear about, please just email me and we'll try and get something set up. Otherwise, we'll just use it kind of as an open discussion time point. So thanks, everybody.